defined at the beginning of chapter 7. Enzyme, activation energy, substrate, active site. Got to know all those definitions. Know that an enzyme functions by lowering the activation energy. Then the figure number 1, the picture of the enzyme reaction that we ran. You need to know that the substrate we used the entire time was lactose, the enzyme was lactase, and the products were glucose and galactose. Not to recreate that scientific diagram, but to know what the substrate enzyme products were. 7.1, we looked at the effects of temperature on enzymatic activity, and after we were done, we ended up with a graph that looked something like this. We used glucose production as a measurement of how active the enzyme was. Most of us ended up with a graph that looked something like this. You need to know what the optimal enzyme temperature was for your class. Most temperature classes, it was 40, 60 degrees around there, but I don't know. Whatever you found in your lab, that's what your lab instructor is going to want. You also need to realize what's happening when it's really hot. The enzyme is becoming denatured. That means that it's losing its three-dimensional structure. That's why it doesn't work so well when it becomes really hot. At cold temperatures, it can't induce fit as well. This entire lab, they ask you what the substrate was, what the source of the substrate was, what the independent, what the dependent variable was. For this experiment, Unit 7, and every other experiment, every single one that we've got, you need to be able to identify independent and dependent variables. Independent variable is always the one that I manipulate. So think I for independent, I was the one who did it. Dependent variable, it's the very end. What was the last thing you wrote? When you were done with the experiment, what did you write down? So D, when you were done with the experiment, that's what you got out. The next experiment, 7.2, effect of pH on enzyme activity. Again, we measured enzyme activity at a variety of pHs from 0 to 12. This graph ended up looking something more like that for most people. It should have been best at around pH 4-ish, but some of your labs do not get this. So if your lab got that pH 7 was the best, then that's what you should know. But know whatever your lab figured out was the optimal pH. It's wherever the numbers were the highest. And then realize again that at the other extremes, the enzyme is becoming denatured. Exercise 7.3, enzyme specificity. In exercise 7.3, we use 2 different substrates, starting things. One time we used lactose, one time we used maltose. You should realize that lactose is going to work a lot better than maltose because enzymes are specific for a given substrate. One way we might ask about this, let's see if we can do this. Tube A has 450 milligrams per deciliter of glucose in it. Tube B has 1,000 milligrams per deciliter of protein in it. Which of these 2 test tubes had lactose and lactase in it? Tube B. Lactose plus lactase, that number would be higher than the maltose plus the lactase. Try it again. Using those 2 tubes, one of them is a pH 12 and one of them is a pH 4. Which one is the pH 4? Tube B. The pH 4 would be the higher glucose production. So that might be another way to sort of ask your knowledge about those. Exercise 7.4 was looking at the cofactors of enzymes. Know what a cofactor is. Cofactors are small, inorganic, usually metallic ions. Things like copper, magnesium, those are cofactors. And they're required to help an enzyme function. EDTA, not a cofactor. EDTA is not helping anything. EDTA is something that we're adding that's blocking the cofactor. So of these 2 tubes, if one had EDTA in it and one didn't have EDTA in it, which one would not have the EDTA? 2B, the one that works. When there isn't EDTA, everything works great. 2A, when you add EDTA, when you block the cofactor, it doesn't work as well. So just realize that interaction. At the very end of chapter 7, we talked through probability and statistics. Realize that every experiment always is looking at 2 different hypotheses. There's a hypothesis that says nothing is going to happen. This is known as the null hypothesis. Whatever you do in group A, it's going to be exactly what happens in group B. The alternative hypothesis says, no, there's going to be a difference. A will not be the same as B. The way to compare 2 averages, sort of like we've drawn out like this, is to do a t-test. When you run a t-test, you can calculate something called the p-value. Know how to interpret p-values. In science, the rule less than 0.05, that means there's a real difference between the groups. Oh yeah, group A was different than group B. If this number is bigger than 0.05, then it means there's no statistical significance, no difference between the groups. Everything from 7, let's go to 8, cellular respiration. The cellular respiration day begins with the chemical equation for cellular respiration. You should know the full chemical equation with all of the prefixes and numbers. I'm going to shorthand it for speeding of time here. But the overall chemical equation for cellular respiration would be oxygen and glucose go in, and then we get carbon dioxide, water, and energy in the form of ATP and heat out. So know that's the overall chemical equation for cellular respiration. In this experiment, we're going to do one experiment where we're going to look at respiration rates by measuring the oxygen consumed. Another experiment, we're going to look at carbon dioxide produced. The oxygen experiment, that's 8.1, that's the mouse experiment. This is an example of a quantitative experiment. There's numbers, you had to do math. The experiment where we measured the carbon dioxide coming out, that's the Bean experiment, that was 8.2. This was a qualitative experiment because it was just colors. We're just looking at the colors. Start with 8.1 and talk about this mouse experiment for a second. In 8.1, at the very beginning of your introduction of 8.1, there's a chart that shows you the relative oxygen consumption rates of various organisms based on size. If we were going to change that to a graphical form, small things like a mouse, they have really high oxygen consumption rates. Big things like an elephant, they have really low oxygen consumption rates. If we make the graph from the chart that they show, what we actually get is a curve that looks something like this. Oxygen consumption rates are always measured in these same units. These units are a little bit bizarre. They are milliliters of oxygen per hour per gram. Those are the standard units. If you're a scientist and you're talking about metabolic rate, you're going to measure it always in milliliters of oxygen per hour per gram. You should know those units. You should know those general ones. The mouse experiment, know what the purpose of the little white pellets was in the bottom of the little container where you stuck the mouse. Those are potassium hydroxide pellets. And what they're going to do is they're going to absorb carbon dioxide that the mice are breathing out. The math on that chart, if I gave you some of the numbers, I would expect for you to be able to do the math to calculate the other ones. Once we switch over to the board and I've got everything shown up, I'll show you that again. But don't think you don't have to do math on this practical. Fair game. So if I give you milliliters of oxygen and how many minutes and the weight in grams, you should know how to change that into oxygen consumption rate. And we'll do that on the board. All right, 8.02, the Bean experiment. Please know that the Bean experiment was a qualitative experiment and that we were looking at carbon dioxide being produced. With the beans, we used a specific kind of bean called a germinated bean. Germinated means alive or breathing. In this experiment, know the name of the glassware. We used those fermentation flasks. The respiration flasks. Also, in that experiment, you should know just outputs, what colors things will turn. We added phenol red. If there is carbon dioxide present, it'll turn yellow. If there's no carbon dioxide present, it will stay red. Exercise 8.3 was an anaerobic process. In 8.3, we stopped doing this thing, the red thing, cellular respiration, and we moved to looking at anaerobic respiration. We looked specifically at alcohol fermentation. Please know that this is anaerobic. It's produced by yeast. And in this process, what a yeast does is it takes glucose in, and by the process of fermentation, it makes 4 outputs. It makes 2 net ATP. It regenerates 2 NAD pluses. It also makes carbon dioxides and ethanol. The way we could tell fermentation was happening is we saw the presence of the carbon dioxide bubbles accumulating. Those other products, however, were still being made, and we don't want to forget that those were there. In fermentation, hopefully we saw 0 fermentation when there was water. We should have seen the most fermentation when we gave the little yeast glucose, and little, if any, fermentation when we use starch. The reason for that being is starch is such a big molecule, it takes a while for the yeast to break it up before it can start doing fermentation. All right, that's everything there. Let's go to photosynthesis. Photosynthesis also begins its unit by rewriting out what the overall equation for photosynthesis is. You should know this, again, with the numbers, the prefixes, etc., knowing the actual chemical formulas. Shorthand way of putting it up, our photosynthesis is we bring in water and light and carbon dioxide, and then what does a plant do? It makes glucose, and a little bit of oxygen as a byproduct. The major site of photosynthesis is the organelle called the chloroplast. And the very first thing we did in exercise 9.1 is we discussed how light works. It went ahead and it relists for you the colors of the rainbow: red, orange, yellow, green, blue, violet. And you should know general wavelengths that are associated with each color. Here are some benchmarks if you have no way to memorize them yet. 700s for red, 600s for orange. Let's skip yellow and green. Blues are about 500, violets are about 400. Green we use a lot in lab, it's right around 540. And so now I'm just going to get something for yellow that's in between 600 and 540. 575 is a nice round number that works. Okay? So in some way, have a way of memorizing it. If your lab instructor gave you a better way or something that you're more comfortable with, do it that way. I mean, they're the ones grading your test. We then talked about why coats are different colors or why is a leaf blue. So my marker right here. Why does it look blue? What is it absorbing? Everything but blue. And so what's being reflected off? Blue. That's why it looks blue. That's always the answer. Don't make it any harder than that. No matter which way I ask the question, that's always the answer. So what we did when in 9.1 was we created absorption and transmission spectra for both spinach and carrot. You do not need to memorize these. Do not sit there and be like, okay, it's 625 absorbance of spinach was. You don't need to memorize those details. It's more the broad spectrum. See if you can do this one. Red, orange, yellow, green, blue, violet. Sorry, my pens are dying at the end of the semester. All right, not much better. All right, what color would this thing appear? If this is absorbance, sorry, I should probably tell you what's on that axis. That's absorbance. What color does this thing look? Green. Green. It's absorbing everything else except green. That's spinach. Okay? So that's all we need you to be able to realize is how to read these charts more than memorizing every single data point. Let's switch this around and change it to this. This is now transmittance. What color is this thing? Orange. Orangish. It's transmitting a lot of orange, not much of anything else. So just know how to read absorbance transmittance graphs. 9.2 was an exercise called pigment chromatography. In pigment chromatography, please know that name. Just like be able to come up with that word, chromatography. You took a little spinach leaf and rubbed it on the piece of paper. When you are done, 4 pigments will separate out. Up at the top, you'll get a pigment called carotene. It'll be orange colored. Then there'll be a second bigger splotch of something called xanthophyll, sort of a yellowish color. And then down at the bottom, you'll have a first green patch, that's chlorophyll A, then a second green patch, that's called chlorophyll B. Please know the names of each of these pigments and the order that they separate out. In addition to knowing their names, you also need to understand the physical properties that govern their movement. So, for instance, carotene, as we talk about it, it is very soluble. It was able to move in that liquid very well, so it traveled very far. Carotene is soluble. On the other hand, chlorophyll B is less soluble. Another way to talk about it, other than soluble, less soluble, is hydrophobic, hydrophilic. Carotene does not have very much polarity to it. It is very hydrophobic. It doesn't like to stick to the paper. That's why it can zoom up so high. And so we say that carotene is hydrophobic. On the other hand, chlorophyll B in comparison is very hydrophilic. It has charge on it. It likes to stick to the paper. You can say it's hydrophilic. 9.3, starch localization in leaves. This was just done as a demo. If your class did not do the demo, go online, watch the video. It's very easy to see the demo. Exercise 9.3, we took a leaf, we had it covered in tin foil, and then we checked to see where there was starch or not in the leaf. Need to know the purpose of some of the steps. After covering it in tin foil, the reason for the tin foil was to make it so that this leaf, when it had tin foil on one part of it, wouldn't be able to do photosynthesis. And so since it's covered in tin foil, what it's going to do is it's going to use up its stored reserves of starch. So that was the purpose of the tin foil. After you pull the leaves apart, the next thing you do is you boil them in ethanol. You do that to remove all the pigments, so you can make a little ghost leaf, so it'll be white. After you boil it in ethanol, you then add iodine. Iodine is added because iodine is a test for starch. After that point, we come back with leaves that look something like this. Where there was no tin foil, they're black everywhere, because there's tons of starch there, and it's just solid black. Where there was tin foil, this part of the plant, you'll see some evidence of starch being there, but not very much, because this is where the plant has used up most of its stored reserves of starch. Things to notice: we used iodine to test for the presence of starch. We learned that last practical. And that starch seems to be localized primarily in the veins. All right, that's good for 9. Let's go to 10. Cell division. Cell division, chapter 10, mitosis, meiosis, begins with a lot of definitions that hopefully you already know from lecture, or if you used to know them from lecture and you forgot them, you might want to revisit them. So we'll begin by talking about the cell cycle. As we look at cell cycle, we often draw it as a big pie chart, and we can divide it up into, I better just draw it the same way I always draw it. All right. Most of the cell cycle is a phase called interphase. That's where a cell is most of the time. When you look at in micrograph, a picture of cells in mitosis or meiosis, most of them are going to be in interphase. That's what you're going to see most of them in. We can divide interphase up into G1, S, and G2. G1 is a growth phase. S is where DNA synthesis takes place. G2 is a second growth phase. This last smaller gap that's left is called the M phase. The M phase takes place of two things: cytokinesis. That's division of the cytoplasm. And then this other part is actually mitosis, division of the DNA. In lecture, you learned a lot about this in more detail, but here in lab for mitosis, we really wanted to see 4 particular stages in really good detail. We wanted to be able to differentiate between prophase, metaphase, anaphase, and telophase. In addition, before mitosis, we would want to know what something in interphase looked like. In general, telophase is pretty easy. There's 2 clumps of something, and they're starting to go back. Anaphase, they're pulling apart. Metaphase, they're lining up. The 2 that students get the most tricked up between with generally is interphase and prophase. So let's go through these 2 specifically. We'll do more pictures once we've got real pictures instead of my bad drawings. Plant cell. We'll do animal cell. That's what you guys did first. Animal cell. When you look at an animal cell in interphase, you're just going to see a circle, and it's going to be pink, and there's not going to be anything you can see at all in it. It's just going to look pink. When you're an animal cell and you go to prophase, all of a sudden, if your marker is not dying, you'll begin to see little funny black things all throughout of it. And a plant cell. During interphase of a plant cell, you're going to see a nice big plant cell, and you're going to be seeing this big gray thing right in the middle. You actually see something. Prophase of a plant cell, you're going to again see that circle thing, but you're actually going to start to see some sort of chromosomes appearing within. Those are the 2 I think that are trickiest. We'll practice them again once we've got real pictures for those of you guys that are sitting in nosebleed seats. All right, other words to know. Know the difference between, I'll go this I think that's good. The other thing that I would know the difference between is cytokinesis, the division of the cytoplasm, whether we're doing it in a plant or an animal cell. When we're in a plant cell, cytokinesis happens by a process called cell plate formation, and the structure that we form is called the cell plate. You have to be really careful when looking at a cell and read the question that's being asked to know whether answering cell plate formation or cell plate, or sometimes the word telophase, would be the correct answer. Because I can show you the same picture and just ask the question different, and I'm going to be looking for a different answer. Rule of thumb. If the question says what phase or stage am I in, answer with a phase or a stage word. Okay? If it says what process is happening, tell me the name of the process. This is cell plate formation. If I ask structure, tell me the name of the structure. That's the cell plate. So just be very careful about reading questions and don't think that you know what they're asking. Make sure you answer what's being asked. Animals is slightly different. In animals, the process is called cleavage. The structure that is formed is the cleavage furrow. And the stage that we're in when that is often happening is telophase. Spend a lot of time looking at those pictures once we get to them. Other big stuff. Exercise 10.3. Meiosis using pop beads. You need to know the 3 things that make meiosis different than mitosis. Those are what you learned in lecture. And homologous chromosomes have crossing over and they pair up during prophase 1. Homologous chromosomes line up during metaphase 1. Homologous chromosomes pull apart during anaphase 1. You need to know what homologous chromosome means. Homologous chromosome is when we look at a cell, there will be 2 chromosomes of the same shape and size that buddy up. So there's this big red guy and there's this big black one. Those are homologous chromosomes. They're the same shape and size. Here's a smaller group. Those are also homologous chromosomes. They buddy up. And we only see that buddying up and lining up during meiosis. Sometimes non-sister chromatids lay on each other and they exchange genetic information. That process is called crossing over. It only happens during prophase 1 of meiosis. Everything else that we want to do that we'll do visually. Biotechnology, chapter 11. Biotechnology begins by reminding us of biology's central dogma. DNA makes RNA, RNA makes protein. You know that central dogma? The very first experiment was the DNA isolation experiment. In the DNA isolation experiment, we took sort of 4 broad steps to isolate DNA. You should know those 4 steps and the purposes of those steps to isolate DNA. So the first step, we blend the cells. That causes them to break open. That's why we blend them, break open the cells. Second step that we do, we go ahead and add meat tenderizer. We add meat tenderizer because that is going to remove the proteins. Third, the next thing we do is we add a soap solution or detergent. That's going to disrupt the lipid bilayer. Fourth, we precipitate the DNA. We make it visible by adding ice, cold, ethanol. Once we precipitate the DNA, you should know what it visually looked like in the tube where it was. It was a little bubbly stuff there at the top. Exercise 11.2, that's where we ran gel electrophoresis. You should know the name of that technique. Little gels. As we run gels, we're going to make an actual jello type thing, a gel, where there's holes for DNA. We can put DNA in the holes and then we electrocute it. When we add in the electrodes, the negative charge is always at the top. The positive charge is always at the bottom. As you add DNA in, pieces of DNA that are really long, we're going to have a hard time moving through the gel, so they'll stay up near the top of the gel. Pieces of DNA that are really teeny will move down toward the bottom of the gel. Once we've ran the gel, exercise 11.3, we can then and take these graphs and just choose a few of the lines to focus on. And when we do it that way, when we're doing the paternity or the crime scene analysis, that is called an autoradiograph. You should know that name, autoradiograph. In a crime scene, look for an exact duplicate between whatever was at the crime scene and whoever your suspect is. Paternity is going to be a little bit different. In paternity, we do something more like this. And, sorry, those of you guys are in the back. If this first lane is mom, and the next lane is kid, and the last lane is dad, paternity analysis, the way you run it is first you find everything that mom has and kid have in common and ignore it. So, mom and kid have this lane in common, this lane in common, this lane in common. I'm ignoring it. Then you focus on what kid has left, and you see if that came from dad. Could this be the dad over here?
No.
No, because I don't have enough bands. This is definitely not dad over here. So that's a paternity analysis. Exercise 11.4 was the genetics and the blood typing experiment. And the blood typing experiment, know how blood type works. We have 3 different alleles to choose from. A dominant A allele codes for A antigens. B allele codes for B antigens. Little I recessive allele codes for no antigens. We can combine these to create a bunch of different blood types. Big I A, big I A. What blood type is that? A. What is it if it's big I A, big I B? AB. What kind of antigens would be on the surface of this blood? A and B. What about this blood type? What kind of blood is that? O. What kind of antigens are on this blood? None. Then you should know about donating and receiving blood. It does not work the same ways back and forth. The people I can give blood to are not necessarily the people I want to get blood back from. The way to think about it is always just think about the antigens. You can only give blood to somebody that they're not going to recognize one of the antigens as being foreign. Let's try a couple different instances. Let's imagine that I have AB blood. That's me. Who could I donate my blood to? Could I give my blood to somebody who had type A blood?
No.
No, because they're going to see my B's and attack that, and that would not be good. That would be a clot. Could I give my blood to somebody who had type B blood? No, because they're going to see the A's and attack it. Could I give my blood to somebody else who was also AB?
Yes.
Yeah, that works. Can I give my blood to somebody who has type O blood?
Yes.
No, because somebody with type O blood is going to see those A and B antigens and react. Let's ask the question the exact opposite way. I have AB blood. Who can I get blood from? Could I get from somebody who has O?
No.
Yes.
Yeah, because if somebody gives me O blood, I'm not going to see anything funny on that. Okay, I have AB blood. Could I get somebody's blood who is A given to me?
Yes.
Yeah, because my body's okay with A's. I can deal with A's. Could I get from somebody who has B's?
Yes.
Yeah, because I'm okay with B's. Could I get from somebody who has AB? Yes. So it's not always both ways. The way we looked at this is we did the blood typing, where it was the little circles, where you had a blood type. Please remember that clumps equal yes, you do have it. So if this is A at the top, B at the bottom, RH over here on the side, and I get clumps in B and no clumps everywhere else, what kind of blood does this person have? B, positive or negative? Negative. It's not positive for RH is the positive one. This would be B negative. It for 11, 12 prokaryotes. Know the 3 different shapes or morphologies of bacteria. 3 different shapes are cocci are the round ones, bacilli are the long ones, and then spirilla are the spirally ones. Please use those actual words. If you just say round ones or long ones or spiral ones, it's wrong. You got to use cocci, bacilla, spirilla. After you looked at the actual shapes, the morphologies of the bacteria, there was a chart where you had to look at the differences between bacteria and eukaryotes, different characteristics. All fair game. You need to know that entire chart. It's 12.2. 12.3 was a Gram staining protocol. The Gram staining looks at the difference between 2 broad types or classes of bacteria. Know the 4 steps of Gram staining. So the first thing that you add is called crystal violet. This is your primary stain. After that, you add iodine, which is a mordant. Third, you're going to add ethanol or grams decolorizer. And then fourth, you're going to come in and add safranin. You should understand the reason behind using each of these stains and what's going to happen. So a gram-positive cell, this is the way it looks. It's got a plasma membrane, PM, plasma membrane. And then on the outside of it has this big, fat wall made up of peptidoglycan. Peptidoglycan has some holes in it, some pores. So in this procedure, what we do is we add crystal violet to the cell. The crystal violet gets all the way in and it's stuck on the inside. We then add iodine. Complexes with the crystal violet makes a big chunk. We then add the third step, ethanol. And when you add the ethanol, it causes all those little pores to close up. We then try to wash the cell. This crystal violet will be stuck inside. It'll be purple. We try to come out